Thanks, Caitlin, for asking me to present today, and I can't express how happy I am that the clinic and this conference is going ahead. It's just a wonderful demonstration of how motivated you all are as a community, and it's great to see how far you've come as an organisation. Caitlin has asked me to speak today about basic scientific and translational research, which I will discuss over the next few slides. But I think the overarching question we want to address when discussing these types of research is really to help families consider whether they want to be involved in research and how their loved ones' samples can be used to advance the scientific community's understanding of DESSH. And this will assist in making sure people with DESSH get the best care in the future as we look towards possible interventions and treatments. Scientific research is a continuum, and it is important to remember that these points, although they are laid out separately, are interconnected, and often we cannot have one without the other. Over the next couple of slides, I want to briefly summarize academic research because that is what you're going to be involved in at Washington University. But I did want to mention the drug development process because these may be terms you come across. And if there is interest, perhaps we can look at these processes another time. Academic research can be split into three types: basic, translational, and clinical, whilst drug development has 5 main stages which leverage off findings from academic research. As I said, although these are annotated as separate points here, it is important to remember that these are very interconnected. They rely on each other, and we cannot have effective clinical advancement and drug development without basic scientific research. Basic scientific research is the foundation of medical discovery. It aims to understand and predict biological phenomena. It is the source of most new scientific ideas and is mostly conducted at universities. Basic science uses cellular components such as purified proteins and genetic material like DNA. It can also use human cells in culture, where they are grown in liquid in plasticware, or animal models such as mice and rats. Basic science is mostly prospective. So we have a question and we design experiments to find the answers. And although the information may not be immediately utilized, it is often an important building block to get to the next steps of our understanding about biological processes. One of the most famous basic scientific research feats in our lifetime has been the Human Genome Project, where in 1990, scientists started trying to sequence the entire 3 billion base pairs in the human genome, and unlocking our genetic code has led to countless developments in our knowledge of human disease, and it will do well into the future. Without the Human Genome Project, we may not know that mutations in the WAC gene is what causes DESSH. Translational research is the process that bridges the gap between taking a discovery from the lab to the clinic and is often referred to as bench to bedside. It takes findings from basic science and applies them with the intention of developing new interventions that will directly benefit humans. And this could be in the form of a new drug, early detection of a disease, better diagnostic methods, medical devices, or alternative therapies. The primary goal of these interventions is to alter events or outcomes, such as reducing morbidity or illness, increasing survival, reducing toxicity, or invasiveness of current therapies. An example of this would be developing an oral form of a drug that a patient can take once a day by mouth from their home, versus having to come into a clinic and get an injection of that drug into their bloodstream. Translational research can use cell cultures, animal models, and human tissues. If we continue with the Human Genome Project theme, it, as a basic science feat, allowed the identification of gene mutation, such as gene mutation X, that causes cancer, that we can now target with a drug to eradicate that cancer in our model systems. Clinical research, as the name suggests, takes the findings into the clinic and applies them to a whole human being rather than tissues or model systems. It explores whether new treatments, medications, and techniques are safe and effective. And clinical research is often administered through clinical trials, which I'm sure everyone here has heard of. This allows for the accurate and precise tracking of patient progress, which not only includes the effectiveness of an interventional therapy, but if it causes any harm to the patient. And these outcomes are reported to and monitored by the FDA. Whilst clinical research, if successful, leads to dissemination of a therapy into the community where it can be implemented as standard of care, which of course is the end goal, this is certainly not the end point. All of these research steps are a continuum, and we can go back and forth between them as more information arises. An example of clinical research in our Human Genome Project example would be to understand whether the new drug that we have developed and tested in our translational research models can extend the life of cancer patients with the mutation in gene X that we were initially able to identify because of the Human Genome Project, a basic science project. So how does this affect you, and how can your samples be used in these types of research? Well, there's still so much we don't know about DESSH and WAC, but the community that you have fostered and your motivation and interest in research has given you such an accelerated start to filling in our knowledge gaps. Just to give you a few examples of how your samples could be used, we could generate cell lines that we can then manipulate without any risk of harm to the patient. This can help us understand WAC function by isolating samples in a controlled environment without confounding factors like what you had for breakfast or how much sleep did you get. We could start to scrutinize the individual mutations in WAC because there have been so many different mutations identified, and we don't know how this relates to which clinical symptoms a person might have, or even if some medications might be better in some patients than others. It allows us to repeat measurements, which is key to validating our findings, and we can also start to modify WAC function. What happens if we knock it out of patient cells, or if we use a compound to block its function if one becomes available? Finally, from my work in other syndromes and in cancer, one invaluable resource is a biobank. Right now, we don't know what we don't know, and we are figuring all of this out prospectively. But if we fast forward 5 years into the future, the landscape could be very different, and having a biobank could allow for rapid investigation into potential treatments. This is not an exhaustive list by any means, but I hope it gives you an idea of what could be done. I'm going to leave this here. And if there's any questions, feel free to pass them to Caitlin and she can send them on to me. I would be more than happy to answer any questions you have. I hope you enjoy the rest of the conference and you enjoy your time in St. Louis. And I wish all of you all the best. Thank you.